in a little earlier, but I think he joined the Center for Computational Systems Biology and Cooperative Agricultural Research Center in 2018 or 19, as to establish plant systems biology research lab. So he has a joint appointment between CCSB and CAIC, has been using state-of-art technology to study plant shooting, branch shooting in plant. So without much further ado, and Dr. Kebrom, it's your turn to give a talk.
Okay, thank you, Dr. Kim, for introduction, and good afternoon, everyone. And thank you for joining the seminar. So the title of my seminar is...
Tesfamichael, I'm sorry, just give me 1 second. I want to start recording your talk today.
Okay.
So I'm trying to find, okay, I can't do that because I'm not a host. Anna, can you start recording the talk?
Recording in progress.
Thanks.
Recording stopped. Recording in progress.
Okay, so the title of my seminar is The Power of RNA-seq Gene Expression Profiling, illustrated by shoot branching research in plants. So I have been doing research on shoot branching since I was a PhD student at Texas A&M University around 2003, 2004. And as you can see, these are plants grown for shoot branching research. You can see this plant without a branch and this branched plant, and this is a field at when I was at Cornell University. So the objective of my research is that one of the methods I used for shoot branching research has been gene expression analysis, and I used it initially when I was a PhD student, Northern blot method. This is Northern blot. And using this method, I was studying one gene at a time. For example, to complete this research, this Northern blot, it takes like 1 month. So to study 2 genes, it took me 1 month. Then I moved to qPCR method, where I can study 10 genes at a time. And now we are doing this RNA-seq, where we can study thousands of genes at a time. So my objective is to show you how this RNA-seq method is powerful, which is available now at Prairie View. And I will start first showing you what I discovered using this and how using RNA-seq my research advanced. And the data I will show you is from these plants. So before going to the gene expression methods I used, I want just to briefly introduce about branch development in plants. So branches in plants develop from buds in the axil of leaves. That's why we call them axillary buds. So plants usually develop in each leaf axil buds, and these buds grow and make or form a branch. So there are 2 types of branches depending on the position of the buds. The aerial branches, what we call them branches, and then in the grasses we observe, or there are branches that grow from the soil surface, and they are called tillers. So these branches, they develop from the soil surface because during early stage of development of these grass plants like wheat, barley, maize, they don't form stem. And the buds are at the base of the shoot near the soil surface. So when the buds grow, it appears as if they are growing from the soil or from the roots, but actually they are branches from the plant. And the development of a branch, the aerial and the basal plant branches or tillers is almost the same, but we call them tillers in general. So shoot branching we study because it has many applications in crop production. For example, the yield or quality of grains or fruits is very much affected by branching. For example, in these plants, as you can see, this plant developed several branches, and their head is very small compared to the ones that developed only one stem or one shoot. So by modulating branching, we can increase yield and also improve quality. Also, this shoot branching knowledge is good for or important for post-harvest handling of crops like potato or root crops, because this potato, for example, it sprouts, and sprouting is a problem. And the sprouting process is from the growth of the sprouts from buds. So knowledge of buds or bud development is also important for post-harvest handling. Also for mechanized harvesting of crops, it is important because some crops, they continuously develop branches, and these branches, they can be at different developmental stage, and it can interfere with harvesting. So farmers, what they do is that they apply chemicals to ripen, to make the ripening process faster, so that the plants can dry and they can harvest. Also for resource use efficiency like water, light, or nutrients. If a plant is highly branched, because most of the branches, they die at the early stage, so they are not efficient at using resource. For example, this plant, it develops about 15 branches, and most of them will die before producing grains. While this plant develops up to 2 or 3 branches, all of them, they produce grains. The amount of water required for this plant is very higher than the amount for this plant. So this is more drought tolerant. So in terms of resources efficiency, shoot branching is also important. Shoot branching research has been done for almost a century. Still, the knowledge required to control shoot branching and increase yield is still lacking. We have only examples of shoot branching modification through crop domestication. For example, in maize, evolved from its wild ancestor teosinte. Teosinte is highly branched, and the number of grains per year in teosinte doesn't go higher than like 12, while maize, it doesn't branch, and the number of grains in the ear of maize could reach up to 500 or more. So it is reduction in branching during domestication increases the yield of maize. Also the same with sunflower, as you can see here. This is a domesticated sunflower, and this is a wild sunflower. This sunflower, wild sunflower, you can see it when you drive along the roads in Texas, still in the wild, and the heads are very small, and that means the yield is very small, while the domesticated sunflower, which is now grown for grain, is as a reproductive structure is very large. So this modern or domesticated sunflower develops only one shoot, so it is highly branched. Farmers also practice manual branch removal, especially these fruit growers, to improve quality of fruits and to increase yield. And this is labor intensive, also cost intensive. So they spend about 20% of the cost of production in manual removal. So the objective of my research is to advance knowledge of the physiological, molecular, and genetic regulation of shoot branching and develop strategies to control shoot branching using genetic means and improve crop yield so that farmers don't need to do manual removal of branching and things like that. Shoot branching research has been done traditionally in eudicots in like beans, peas, or Arabidopsis, but I did my research. I started with the grasses, the major crops, because these are very important crops. Most of our caloric needs are from these crops. They are grown also all over the world, so there is a lot of diversity in these crops like wheat, sorghum, or maize. So that's why I continued my research, although it's challenging because we don't see the buds in these crops. They are covered by the leaves, but I developed a method of harvesting and studying these crops. So I did, and I am doing still my research on the major grain crops. And I did research in sorghum at Texas A&M University with these plants, and at CSIRO with wheat, and on maize at Cornell University. So first I will briefly mention about branch development. Branch development starts with the establishment of axillary meristems, a few meristematic cells or stem cells capable of dividing are formed at the base near the leaf axil in the stem. Then through division, these meristematic cells, they form a bud. So the bud is formed and it's now pushed away from the main shoot axis and it's sitting here on its own. So once it is formed, it enters a stage called the transition stage. And we call it transition stage because at this stage, we can change the fate of the bud. We can induce it to be dormant or grow. So it enters the transition stage, but we don't know what goes on in this transition stage, but I will show you later that through RNA-seq we identify this. So once a bud is formed, its dormancy or growth is regulated by plant hormones and environmental factors. And some of the hormones that control the dormancy or growth of buds are cytokinins, auxins, trigolacton, and abscisic acid. Sugar is also sometimes considered as a hormone, which some people think, but it may not be. It is, I think it is a source of energy for the growth of the bud into a branch. So cytokinins, they induce the growth of the bud when they are directly applied. This is more frequently, but not always. Auxin and strigolactones, they are synthesized in the shoot tip. Auxin is synthesized at the tip of the shoot, and strigolactones in the roots. And both these hormones indirectly, without entering into the bud, they induce bud dormancy. Abscisic acid is another hormone that induces bud dormancy. Actually, its level increases in dormant buds. That's why it is called a bud dormancy hormone. Environmental factors that control shoot branching include water, nutrients, and shade signals or light. Nutrients like water and fertilizers, they usually also affect the growth of the whole plant, but shade signals, they promote other developmental processes and inhibit shoot branching. So my research has been on shed signaling for the past 15 years. So how, in this seminar, I will show you what I have done with these sorghum plants on shed signaling, and using northern blot first and then later RNA-seq. So how does shed affect this bud development or growth? So plants, they have photoreceptors for monitoring the level of light, the visible light, from around 400 nanometer wavelengths, the blue range, up to the far red around 780 nanometer. So for the blue, they use a photoreceptor called cryptochrome, while for the red and far red, they use phytochrome. And phytochromes are the ones that monitor shading. So these phytochromes, they absorb the red or the far red. So when they absorb red, which is important for photosynthesis, they become active. When they absorb far red light, which is a kind of shed signal, they become inactive. So if you put a sensor under a leaf, the red is absorbed by the leaf for photosynthesis, only the far red is transmitted. So a plant that is shaded perceives the far red because the red is being taken up by the leaf over it. So if a plant knows that, or using these photoreceptors recognize that it is shaded, then one thing it does is that it stops branching. And a plant that is mutated with these photoreceptors, it is like always, it thinks always shaded, so it doesn't develop branches. So this plant identified a long time ago and being used, that has been used by scientists at Texas A&M University for shed signaling research, is it doesn't have the photoreceptor phytochrome. So the phytochrome is mutated in this plant. The plant always thinks shaded and it doesn't branch. While this plant is a sister line, they are genetically, they are very similar, except this one has a mutated version of the phytochrome B. So this plant has normal Phi-B, so it responds to the light. When it is shaded, it becomes like this plant. When it is unshaded, it branches and continues its normal development. So when I started my research based on this knowledge, I developed this model. This is around 2004. So the model is that when there is red light, phytochrome B is active, so it promotes shoot branching. When there is far red light or when the plant is shaded, then phytochrome B becomes inactive, so shoot branching is inhibited. So I used this model to start with my research. So during my PhD, there were very few genes known to control shoot branching. And among these very few genes, the two genes were the DRM1 gene, dominance-associated gene, and the tyrosine branched 1 gene. So DRM1 was first identified in P in 1998 by Staffstrom and his colleagues. And this gene is expressed in dormant buds. It's not expressed in growing buds. It's only expressed in dormant buds. And its function is still unknown, but it is a good gene for using it as a marker. So what it means is that if I have a bud, then by studying the expression of gene, I can tell whether it is dormant or growing without any visible sign of growth or dormancy. So I identified this gene in sorghum, and I used it as a marker for my research for dormancy. The other gene is a tyrosine branched 1 gene, And this gene was identified first in maize in 1997 by John Dobley. And John Dobley is a professor at University of Wisconsin, and he's an evolutionary biologist and a genetist, and he studied the evolution of maize from teosinte. Teosinte is a wild ancestor of maize, and maize evolved from teosinte through mutation in about 5 major genes, and one of the genes is TB1, and TB1 is expressed in the buds. It encodes a transcription factor. That means its function is to regulate the expression of other genes. And this gene, during domestication, it was mutated in maize, but the mutation is a gain of function mutation, which means that it becomes, it's the way it functions is different in maize than in teosinte. In teosinte, its level of expression is low, while in maize, its level of expression is high and at all time. Nothing can change its expression level. Because of that, the buds in maize, they don't grow. So maize doesn't develop branches. While if the TB1 in maize is mutated, now loss of function mutation, then the maize plant looks like teosinte. So this is a maize plant with a mutated version of the TB1 gene. So when I started my PhD, I hypothesized that maybe shed signals control or phytochrome B control shoot branching through inactivating this gene in sorghum, because genes are conserved in different species. They exist almost in all plant species. So TB1 now is known to function in almost any plant species studied to date. So what I did is that I developed a method of harvesting buds from sorghum, and I can measure its length and extract RNA and study the gene expression. So what I did is, when I started this research, because research on shoot branching in the grasses was not common because the bud is covered by the leaf. So what I did is that I grew a large number of plants and I did this destructive sampling. That means that I harvest a plant and I remove the leaf. And here, after removing the leaf, you can see the bud. And I pick the bud with a needle and put it on so this bud is microscopic. It is less than 1 millimeter in length. So I use a microscopic meter, this meter, which is about 2 millimeter long. And I put the bud on the slide. This is a slide. And I look at under a microscope to measure its length. So this is James Schnabel, a biology major student at Cornell University, who was working with me. He's dissecting axillary bud. So what I did is that I grew these plants, the wild type, the normal plant and the mutant, and harvested buds at 7, 8, 9, 10, 11, and 12 days after planting. The bud in the first leaf axil, this one. So as you can see here, at 7 days after planting, the length of the bud in the wild type and 5B, the mutant and the wild type, were comparable in length. After that, the bud in the 5B mutant stopped growing, you can see, while in the wild type grew and formed a branch. So what I did is that I harvested RNA from the 5B1 and the wild type at 7 and 9 days after planting, and I studied the expression of TB1 and DRM1. So this is RNA from the 5B1 at 7 days, from the wild type at 7 days, RNA from the 5B1 at 9 days, and from the wild type at 9 days. And this gene is ubiquitin gene. It is expressed in any plant tissue at a higher level. So what we do is we use this plant as a loading control. For example, the difference between these two samples, if it could be difference because the amount of RNA I use, but using this gene, I can see whether the level of RNA I use it were equal or not. So this is a loading control gene. So as you can see here, at 7 days after planting, the TB1 gene was higher in the 5B1 that stopped growing compared to wild. And this is consistent with its function. And at 9 days after planting, the level of TB1 was high. So the dormancy could be associated with a level of TB1. And the DRM1, the dormancy marker, at 7 days, it was low. And as you can see, the bodies were, their length was almost the same. But at 9 days, it was upregulating, indicating that the bodies in the 5B1 in this mutant became dormant at 9 days after planting. So to confirm this, I treated this plant with shed signals. That means I applied shed signals. So the experiment was in a growth chamber, and these are the control plant, and these are the shed treated plant. So the light coming from the ceiling, from the top, was the same on both. That is a photosynthetic light, while I gave to these plants additional far red light, starting at 7 days after planting when the buds were formed. So this is in this plant, okay? So what happens is that at 7 days in the control, the length of the bud was less than 1 millimeter. At 9 days in this control plants, it reached about 4 millimeter in length, while in this far red treated plants, the length was 2 millimeters, so the growth was suppressed. So as you can see here, the expression of the TB1 was high in the control at 7 days, then it was reduced because the buds started to grow. But in the far red treated, it remained high. And the dormancy associated gene, it was upregulated in the far red treated buds or plants, and this indicates that the plants became dormant. So this result shows that the TB1 is involved in regulation of shoot branching by shed signals. And based on this, I modified the model from this one that I developed initially before I started the experiments to this one. So here, what the model is that high red far red or red light activates 5B. 5B inactivates TB1 through unknown mechanism. We don't know the mechanism. There may be other genes between 5B and TB1. And then when 5B inactivates TB1, the buds are free to grow. If the plant is shaded, then 5B becomes inactive. TB1 remains active, and then the buds become dormant. So this was a model around 2006 or 2007. Then from this, this is by qPCR, but I will go directly to save time to the RNA-seq study. So I shifted to using RNA-seq. This study was conducted around 2015 when the RNA-seq technology became common and well developed. So what I did is that I harvested buds from 5B1 and wild type at 6, 7, and 8 days after planting. So earlier study was at 7, 8, and 9 days after planting, but this one was at 6, 7, and 8 days after planting. And this is because I started using a new growth chamber, and when I changed the growth chamber, because of the model or the settings in the growth chamber, the growth of the plants could be shifted by about 1 day, either delayed or it could be earlier. So the study was conducted at 6, 7, and 8 days after planting. So I harvested buds from 3 plants. 3 experiments in each. So at first I grew plants, then I harvested at 6, 7, and 8 days after planting, buds from 5B1 and wild type. And I repeated the same experiment again and again 3 times at different times. So these are from 3 samples from 5B1 from wild type, and the total RNA-seq reads. That means the total RNA fragments that were sequenced were 41 million from 3 biological replicates in the 5B1 at 6 days, and in the wild type about 34 million, and in the others also this was a number of reads. So we generate a large number of fragments and sequence. Then we mapped the fragments to the sorghum genome. By this time, the sorghum genome was sequenced. So about 93% of the RNA fragments sequenced from the 5B1 bud were mapped to the sorghum genome. On average, about 90% were uniquely mapped. That means mapped to unique genus. And about 4% were mapped to non-specifically, and about 6.2% were not mapped. So this means, although the sorghum genome is sequenced, it is not 100% complete. Some genes are still not yet sequenced. That's why it's less than 100%. So I used this principal component analysis clustering method, and I look at how the samples from the 5B1 and wild type buds were, as RNA were clustered. And as you can see here, at 6 days, the wild type and 5B1 buds were clustered together. And at this stage, there was not that much difference between the two. And the number of differentially expressed genes were around 130. At 7 days after planting, they start to slightly differentiate. These are the samples from the 5B1 bud, and these are from the wild type at 7 days. And the number of differentially expressed genes were 350. It increased or doubled, and this is because now their fate is different, going different way. And at 8 days after planting, these are the samples from the wild types, the buds that are going to grow into a branch or branches. And these are the buds from the sample from the buds of 5B1 that became dormant. Now you can see the number of genes differentially expressed between the two. Out of about 40 genes was 611 genes. And we did this Venn diagram, and what we found is that about 71 genes were common between the three. So out of 127 genes that were differentially expressed at 6 days after planting, 71 continued or were also differentially expressed at 8, 7, and 8 days after planting. So there is a tool called MapMan that categorizes the differentially expressed genes and gives them or categorize them into their function, different function. So MapMan has 35 categories for different functional categories. For example, if the gene is one of the differentially expressed gene was at 6 days, it's involved in photosynthesis, so it categorized as a photosynthetic gene. You can see hormone-related genes were upregulated. 3 of those 130 genes that were differentially expressed at 6 days after planting were upregulated, and 5 were down in the 5B1. So almost half of the genes differentially expressed were upregulated, that means turned on, and about half were turned off. So not all genes were turned off or turned on, but some of them were activated because they are required for dormancy, because the bud in the 5B1 becomes dormant, and half of them were required for growth, so they are not needed, so they were turned off. The same as 7 days, almost about half were upregulated and half were downregulated, and at 8 days, almost around half were upregulated and half were downregulated. Then we looked closely at some of the genes, and these are the hormone-related genes that were differentially expressed in the 5B1 buds. And the red ones are highlighted ones are the ones that are downregulated, and the green ones are the ones that are upregulated or turned on. And some of the boxes, as you can see, are not highlighted. This means the gene was not differentially expressed at that time point. So as you can see here, cytokine-related genes were differentially expressed, and this plant hormone is required for bud outgrowth. And as you can see here, genes that are induced by cytokinin were downregulated, starting at 6 days and then at 7 and 8 days. And this green highlighted gene called cytokinin oxidase is a gene that encodes a protein, or that makes a protein that deactivates cytokine. So this gene was upregulated in the 5B1 buds. So this means the cytokinin level was reduced, and these marker genes are telling us that it was reduced because of this gene becomes active. Abscisic acid-related genes were also differentially expressed, but not the way we expected. So studies in Arabidopsis identified that abscisic acid is the one that functions in shed signaling and shoot branching, but it doesn't seem because the gene they identified was NSAID3. This gene is required for abscisic acid biosynthesis. It's a key biosynthesis gene. If there is no, this gene is mutated, then the plant doesn't make abscisic acid. So what they found out is that this gene is upregulated by shed, but from the studies that we conducted, it looks like this is not the earliest gene. The genes that we found, or the genes that we found to be differentially regulated were cytokinin. Like I said earlier, this RNA-seq also surprised us with many things that we didn't expect. For example, ethylene is a plant hormone. It's not known to function in shoot branching. It is a hormone that is involved in fruit ripening and seed germination as well. But we found that a key biosynthesis gene for ethylene was downregulated in the 5B mutant. So this result is indicating that this ethylene is also important in shoot branching, but still there is no study on ethylene and shoot branching. Auxin, although there were some differentially expressed genes, but it was not kind of important for shoot branching related functions. So the most important discovery about hormones from the RNA-seq study was about cytokinin, and I will show you how important this is. Another result that we didn't expect is that differential expression of flowering-related genes. So in the buds, flowering-related genes were differentially expressed, and these genes are very important in the transition of plant from vegetative to flowering. These genes include the terminal flower 1, leafy 3, and apetal 1. These are key genes that control flowering. They were differentially expressed, and this was a very interesting result, and that needs further research. And also another hormone which is involved in flowering was also genes involved in that, and the regulation of that hormone were differentially expressed. And these genes are the GA2 oxidase genes, and these genes activate this flowering promoting gene. So what we observed is that flowering inducing genes, genes that inhibit flowering were activated in the bud in the 5B1. So probably it is to prevent the bud from transition to flowering before it makes branches. Another important discovery from this study was the expression of genes involved in the trehalose pathway. Trehalose is a sugar molecule that is synthesized in a very low amount in plants. There are few labs that can measure this trehalose sugar. And genes involved in trehalose pathway were differentially expressed. In particular, this gene, the TPPI gene, this TPPI gene was upregulated in the 5B1 bud, which means it is required for bud dormancy. And interestingly, I didn't know this TPPI, the name is from Arabidopsis, is a similar gene in maize known as Ramosa gene. Ramosa means branching, and RMS, this gene is the maize gene RMS3. So I didn't know, but recently when I was discussing with Dave Jackson, he told me that this gene is the gene that he identified in 2006 in his lab. And this gene in maize, it induces, it inhibits branching of the tassel. So this was another thing that is important discovered from this study. And this study was published in 2016. And since the publication of this study, there are several papers about trehalose and shoot branching that are published and citing my paper. So what was the most important discovery also from this is about the transition stage. Until recently, we didn't know about what goes on at transition stage, but this RNA-seq data from this sorghum plant gave us some insight, new insight into the transition stage. So I will show you this in more detail. So as I have shown earlier, at 6 days after planting, the buds in 5B1, in this one and this one, are comparable in length, and the expression of the dormancy marker gene was low in both. Then after 6 days after planting, the bud in the 5B1, in this one, stops growing, and the expression of dormancy associated gene or the dormancy marker gene was upregulated, and telling us that the bud became dormant after 6 days after planting. And from the cytokinin data that I showed you, the cytokinin inducible genes were upregulated in the wild type, the bud that grows into a branch. And this is also clearly shown by this, some of the data from the RNA-seq. While in the 5B1, cytokinin responsive genes didn't increase or were not upregulated. This means the level of cytokinin in the 5B1 bud was low. And this is because of this deactivating genes. The cytokinin in the bud was deactivated. And another group of genes that we found was cell wall invertase genes. 2 cell wall invertase genes were upregulated in the 5B1 buds, the buds that grew to a branch. And in the 5B1, these genes were, these 2 genes, they are the same genes, were not upregulated, which means they were very low and they were almost zero. So this is telling us that cell wall invertase genes were upregulated in the growing bud after 6 days. That means during the transition stage. And the function of this cell wall invertase genes was to import sugar. So one way a plant imports sugar is from its surrounding. So first sugar flows to the space around the cell, and these cell wall invertases are on the cell membrane, and what they do is that they chop the sugar so that it can be converted. It will be converted into its simpler form that can be transported into the cell. So the growing bud activated those genes, while in the dormant, the buds that will become dormant, they were not activated. And the other gene that was differentially expressed was asparagine synthase gene, And this gene, it functions in sugar metabolism, and it is always activated when a cell is starved of sugar. A cell, if it doesn't have sugar, this gene goes up. So as you can see here, after 6 days after planting, in the bodies of the 5B mutant that stopped growing or that became dormant, this gene was upregulated, indicating that the bodies were starved of sugar. They had no sugar. So based on this result, we developed this model. So once the seeds are of wild type and 5B1 plants are germinated, the buds in the first leaf axis develop and are until 6 days after planting. This is a bud formation stage. Then the buds enter a transition stage. During this time, the level of cytokinin and sugar in the 5B1 bud was very low, and that the bud became dormant. So that bud transitioned into dormancy. During this time, also in the wild type, that of the bud of the wild type that grow into a branch, the level of cytokinin was high, and the level of the sugar importing mechanism was also activated. Then the bud grew. So this sugar importing mechanism is known as the apoplastic sucrose import mechanism. So apoplast is the free space of in the plant body. So how does then, the question is, how does then a bud is formed? Why the sugar was available during bud formation? So during this stage, the bud is part of this stem, and cells in this section of the stem, they are connected to each other through a tube called plasmodesmata. So sugar flows from one cell to the other cell through a tube. So during this time, maybe the buds were part of the main shoot, and they were connected to this symplastic system, and they were getting sugar. But after they are formed, they are pushed away from the main shoot axis. So now only they can get sugar through that flows into the empty, or the empty space. That means through the apoplastic system. So in the 5B1 buds, the buds didn't get maybe this sugar, or they didn't activate the apoplastic system. So that's why they are becoming dormant. So until during formation, they used the symplastic sugar, while during growth or dormancy, they have to use either apoplastic sugar or they become dormant. So now our model was this around 2007. By 2016, our model now became even better, because now we know what goes on at transition stage. So what happens is that high red far red activates 5B. 5B deactivates cytokinin oxidase. And cytokinin oxidase, if it is not in under shade, if it is the plant is shaded, there is no 5B to deactivate cytokinin oxidase. So this one inactivates cytokinin. But if 5B is active and the plant is not shaded, then cytokinin oxidase is deactivated. And this knowledge is from this paper published in 2007 by a group that study shade signaling and plant development. Now, what does cytokinin do? Cytokinin is, they activate another genus called A-type response regulators. These genus are activated by cytokinin, and this genus activates cell wall invertase. So what happens is that cytokinin activates the sugar importing mechanism. Once sugar is available in the bud, then we know that sugar inactivates tyrosine branch 1 expression. So when sugar inactivates tyrosine branch 1, then the buds grow. So we know also that in some plants like sweet corn, even if the teosynth branched one is expressed at a very high level, the buds still grow, which means sugar may override the teosynth branched one and induce bud outgrowth. So this is our model. So this was by Northern blot, one gene at a time, this model, but using RNA-seq, we now have this model. So to summarize, RNA-seq is a powerful tool for plant physiology and development research, and the tool is now available at the Center for Computational Systems Biology, and we have also the computational tools. So if you are interested, you can do RNA-seq. So thank you if you have any question.
Can I ask a question, Dr. Kebrom?
Yes.
Okay, I'm not a plant physiologist or geneticist and know nothing about it, but from practical approach, once you mutate a plant from the one that has a lot of shoots to a one that has lesser number of shoots and it produces higher quality, better yield of the crop. Now for the next season, when you use the mutated seeds or the seeds from the mutated crop, next season when you plant them, will they give the qualities, characteristics of the mutated plant, or will it give the characteristics of the father, if one generation?
So yeah, so what it means is that the plant in its genome, in its DNA, the gene that controls the growth of branching is mutated or some other processes. So it is always inherited.
Also, it's once you mutate a seed and get a mutated plant, the seeds from the mutated plant will always stay muted.
Yeah, it is in its genome.
Oh, okay, so it's once you do it, that a success is forever,
yes.
Not once.
Yes, yes.
Thank you.
You're welcome.
Hi, Tesfamichael. I do have a question.
Yes.
So I understand that you are mapping out the very basic processes that are controlling shoot branches, and you've made some success.
Yeah.
Do you have any idea how you could translate that to affecting shoot branching in commercial crops?
Yes. Now we have some idea, but we have to test it. So we have several candidate genes, but so I have been doing also shoot branching in response to other factors. So the preliminary data is that the genes that we found in this shared signaling, some of them, they don't function in, for example, other processes. So we have to first confirm that, or we have to first identify genes that function under all environmental and hormonal conditions. These are the ones that are useful. Not otherwise. We have a lot of now we have the knowledge what to do, but we have to test it.
I see. And
Yes.
Is there, like, can you spray hormones or something like that on them?
Yeah. For example, if you spray cytokinin, you can promote shoot branching, but not always. So that is a problem with this shoot branching research. We have different pathways, but we don't know where they converge on. What is the main downstream genes? What are those? That's our objective now.
I see.
But you can spray hormone and either promote or inhibit branching. But this is an indirect mechanism and it doesn't work always.
Thank you very much.
You're welcome. Good question.
Any more questions from the audience? if not, Tesfamichael, I just asked, what is, where are you going from here? I mean, in terms of, one of the reasons you are here at Tradeview is to kind of work both with computational people like us and the plant science people at CRC. So what is it your plan to actually take advantage of such opportunities?
Yeah, so first, the shoot branching research needs. So for example, we are doing in wheat, we have a different process that controls shoot branching. In maize, there is a different process, so we have to first do a lot of RNA-seq and try to figure out which ones are the common genes that control shoot branching. And the other thing, so this is one project that should continue, and that's why we got the funding for the wheat, and even that is at initial stage, but this is one thing. The other thing is that the shed signaling itself, now we are studying how shed controls not only shoot branching, but flowering time, which is important also in shoot branching as well as in other processes. Stem elongation, which is stem is a main yield of for biofuels. The sugar is stored in the stem, and also the stem itself is storing sugar. So those are the things that we should continue doing this RNA-seq study to identify those genes that control flowering, stem development, shoot branching, and other processes as well.
Okay, so the another part of my question was how the computational tools that are available to you from CCSB is going to help you do what the research that you just described.
Yeah, so when I generate a lot of data, RNA-seq data, and from different species through different molecular mechanisms and physiological processes, then the tools available at CCSB and the skills will be very helpful to identify the genes. For example, the method developed by Dumitru, this genome fabric paradigm, seems to be very interesting to identify which group of genes are functioning together to control shoot branching and other developmental processes. In the past, I was doing, so for example, this RNA-seq data that is published in Plant Physiology that I showed you, I did all the bioinformatics, computational, I did all myself. The second author, Dr. Mulat, is a genetist, and I did everything myself. But working with computational biologists who are focused on developing these computational tools is an advantage because on daily basis, they follow the development of the technology. Me as a plant scientist, I grow a lot of plants, and sometimes I lag behind on this new technology, so they can help facilitating my research. That's what I think. And it is always advisable for plant scientists. They should know the computational tools, understand how it works, and also the